Hi, year 13s. I Unfortunately, my recording that I made in class did not record, so I'm doing this again. So today we're going to start our section on selective breeding. Now, traditionally, a farmer would look at an animal and see the phenotype that they liked. So, in other words, if you're a Holstein cattle farmer and you see that there is a bull that produces female offspring that has really big udders and very good milk production, then you're going to keep on breeding with him because you're looking for the phenotype of large udder or something like that. So the problem with that, and specifically when we refer to BLAD, the BLAD mutation in Holstein cattle, and as you know, we have a lot of Holstein cattle in New Zealand as well because we depend heavily on the milk industry for our economy. Um, if you only focus on the visible phenotype, there might be some recessive phenotypes that are accidentally selected for, and you are increasing the prevalency of this recessive phenotype in your herd. And when I say the herd, I'm talking about the whole country's herd. And that obviously is a problem. So to get around that, our modern techniques of selective breeding is to actually analyze the DNA of the organism to see if we want to breed with that specific individual. So if we look at the picture of this cow right in front of us, how do we know if they do not have the recessive mutation that causes BLAD? Um, BLAD is Bovine Leukocyte as Adhesion Deficiency, and we'll go into a little bit of detail about that. And it's a lot of big words, but could have been anything. Okay, it could have been that her milk that she produces is green, and we don't want that. Okay, so how would we know to put all the money into raising a calf until she is fertile and then can actually have babies? And when she has her first baby, then her milk production comes in, and then we can only then find out that she produces milk that we don't want. So it's a lot of time wasted and a lot of expense wasted because you could have had a a producing cow that is going to be economically a lot more viable for you. If only you did the genetic analysis of them. So this is becoming more and more prevalent, and specifically with things like artificial insemination. So they check the genomics of the sperm donor, and then they would know the genomics of the female cattle because they would see the offspring and know if they have. And if they really wanted to, then they can actually check both the male and the female genetically to see if they have the desired trait or if they don't have the undesirable trait. I hope that makes sense. Okay, so what is BLAD? Now, this is not um, compulsory to do BLAD, but BLAD is a good example of where they used molecular techniques for selective breeding. So as you can see here, this person has stepped in a nail, okay, and there's an injury. And when there's an injury like this, we have leukocytes that are big white blood cells, and they are phagocytes. So the leukocyte comes and then they phagocytose the bacteria that you would see over here. They take them, they engulf them, and this is the opposite of exocytosis. So they engulf them, and then they're basically in a type of vesicle or a vacuole, but this time it's called a lysosome. And then into that lysosome, you would secrete um, enzymes that will, like lipase, that will dissolve the membrane and so forth. So in that little vesicle that's going to create it, there's a lot of chemical warfare that goes on to kill bacteria. And this is how your immune system, that consists of your white blood cells, fight infections at a wound. Now, if when there is an infection, your white blood cells are in your blood vessel, and they need to know where to get out. Okay, and the way that they get out we'll go into in more detail on the next slide. But that is the problem with BLAD. BLAD is bovine leukocyte adhesion deficiency. So it's a genetic recessive disorder, and as I said, the leukocyte is a type of white blood cell. So when we have an injury, the epithelium, or then the cells that make up the wall of the vessel, will grow these, I think we call them receptors, and on the leukocyte itself, it already has these membrane proteins that stick out. And think about it like biological Velcro. Okay, so this guy is rolling along in the capillary, la 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 la, and eventually he gets to one of these, and suddenly there's now hooks that catch his proteins that's on his membrane, and that is called adhesion. And when he gets adhered to that area, he stops moving, or she, and when the leukocyte then stops moving, it will then go through the wall of the vessel, because these receptors on the vessel will only be there if there's an injury nearby. And it was through chemical signaling that they know to grow these. Okay, so that's the adhesion part. So if you have bovine leukocyte adhesion deficiency, it means that it doesn't adhere. And then they don't know where to go out. So this disorder is a genetic disorder, and as you can see, the membrane proteins that sit there, that are the ones that attach to the proteins from the blood vessel, they do not exist, and consequently the white blood cell never is stopped, and therefore it never leaves the vessel, and because it never leaves, it never kills the bacteria. So when these sufferers of leukocyte adhesion deficiency have an injury, and this happens in multiple different organisms, including humans. But if they have this deficiency, then they suffer terribly from wound infections, and they don't have a very long life expectancy because nothing goes and fights the infections. Okay, so that's BLAD. Now, BLAD is recessive, and because it's recessive, the farmers using traditional methods did not know that this even exists, because can you see if the organism carries it? No, because remember, you can only see a recessive phenotype if it is homozygous for recessiveness. And in this case, the disorder is on the surface of the leukocyte. So can the farmer, the traditional method, can he see that? No, he can't. But he can see that the babies that are born die very quickly because their wounds become septic and they don't make it. So we don't want this recessive gene in our herd. So therefore, we're going to check if this gene is in the sperm donor, and also if it is in the female. Okay, so the disease can be hidden in heterozygotes. So again, traditional selective breeding. You can only choose what you can see. So if you were a milk farmer, would you choose this cow over here to have babies, or would you choose this cow over here to have babies? And hopefully you all said you want the one with the big udder because it produces a lot of milk. So you will choose her. But how do you know if she is a carrier of BLAD? And consequently, we need to check the DNA. So to check the DNA, there's a few techniques that we can discuss. The first is marker-assisted selection. Now, there's a lot of big words on this page, and don't get confused, because remember, we are talking about using genotypes for selective breeding. We are not going into the genotype and changing it like we do in the other, in transgenesis. Okay, so when we look at marker-assisted selection, the marker is something that points to the gene being present. So marker-assisted selection is a process of DNA markers as indirect selection criteria for selecting agriculturally important traits in crop or stock breeding. Okay, so it's an indirect selection. So think about it as if you're doing, I don't know, geocaching, or you're doing orienteering. You need to find the marker. And at the marker, you're going to find the gene in question, whether that's a positive gene or a negative gene. And that marker, where we have this, it says that the gene is present. And if the gene is present, we call that procedure the quantitative trait loci. Again, new words, but quantitative simply means, is it there or not? Trait refers to the phenotype. In other words, is the gene, so this one, is the gene there that causes BLAD? And if the gene is there and it causes BLAD, where on the chromosome is its location? That's what loci mean. Okay, last year you learned about crossing over and you learned about the location or the loci of the genes. So again, we call this QTL. So you have marker assisted at the top. Underneath it, you have quantitative trait loci. So in other words, is the marker there? And does the marker point to the trait? And where is it? Okay. And if you run a gel and you are looking for a specific trait, you can see there's, for instance, if this was one that we wanted or not wanted, then these three individuals will be excluded from our breeding program. Okay, this is fast. You don't have to wait for maturity. So let's think about if you're a apple breeder. So if you're an apple breeder, it takes a few years for an apple tree to produce its first apple. And let's say there's a new fashion and everybody wants apples that are now red on the inside. So the first thing you do, the scientists will go and they will find which gene causes the red apple flesh. And by doing that is quantitative. You take a seed and you take 1 or 2 cells from the seed, then you get the DNA out, you do PCR, and then you say, okay, quantitatively, is the gene here? Yes or no? Will that gene cause the red flesh? Yes or no? And then where is it? Because we don't always want to run the whole genome. Because for you, if we want to every time map the whole human genome, we're looking for something, it takes days to map a single genotype of humans. So we want to know where it is so that we can actually zoom in and not spend as much time to find these things. Okay, so the definition of QTL, quantitative trait loci, are genetic reasons that influence phenotype, variation of complexity of a complex trait. So it influences phenotypic, in other words, how I look, variation of a complex trait, often through genetic interactions with each other and the environment. Okay, so to put it simply, as I said before, quantitative means can I count it? Is it present? Trait means the phenotype. Remember, phenotype is physical feature of the phenotype. Think about the alliteration. And the loci means the particular place where a gene is found on the chromosome. So you put it together. The place on the chromosome where we find the gene that this particular phenotype is for, if it is present. Okay. So if you look at Holstein cattle, if the individual has the gene for the trait that will produce BLAD, the disorder, then you will not breed with that animal. So to check if the gene is there, it's called the QTL. Okay. So think about it. We have salmon, okay, and baby salmon are called fingerlings. And if you want to see if you're going to invest all the time and money to grow and look after a salmon until it's large, if you're a salmon farmer, and you wait on, let's say, a year or 2 years, and then you have to cut it open to see what color its flesh is. That's not ideal. But if you take a fingerling and you take one of its scales and you can extract the DNA from it, or one of its cells and extract the DNA from it, you're not hurting the fish. But then you can see, does it have the desirable red flesh, or does it have the undesirable pale flesh, because consumers have decided that they want the red flesh. It looks healthier, it looks fresher than the pale flesh, even though there's no difference between them. So salmon farmers want the red flesh. Okay, so they can easily see it without having to either hurt the organism or without having to wait very long. So these genetic markers, so how do we test if we have a QTL? A genetic marker is basically a flag. And as a flag you put up to say, hey, I've got the red flesh gene if I'm a salmon. Hello, you should definitely raise me, I've got red flesh. Okay. If you're a Holstein cattle and you say, hey, I've got the BLAD disorder, please don't breed with me. Do you understand? So if there's a marker that we look for, so instead of looking for the whole gene on the whole genome, we just look for the marker, because the marker is associated with the gene. Sometimes the marker is inside the gene, sometimes it's outside of the gene, but the marker is associated with. So if the marker is there, it means the gene you're looking for is there. And remember, that gene we want is the QTL. Okay. So we look for markers, and there's two types of markers. A single nucleotide polymorphism, SNP, and I know more big words, and we'll unpack that in a moment to make it simpler, or a longer sequence, such as mini and microsatellites. Okay, a satellite is something that orbits a planet, but in this case it orbits, not really, it is around or somewhere close to the gene we want. Okay, so these genetic markers play a key role in genetic mapping, specifically in identifying the positions of different alleles. So let's look at single nucleotide polymorphisms. Now in BLAD, bovine leukocyte adhesion deficiency, the mutation is a single nucleotide polymorphism in the CD18 gene. Now that's just the gene's name, okay? Don't worry about that. But it's a single nucleotide. So literally an A for adenine was replaced with a G. That's all that happened. One base, one nucleotide was changed. And because A binds to T in the DNA and G to C, it changed the whole gene. And because that has then, if you look at it here, there's the normal sequence. So it used to be GAC, which forms our codon that will code for aspartic acid, which is the amino acid. And for those of you who didn't do protein synthesis, we always have, our DNA is always read in threes. We call them triplets. And then on the RNA, it becomes a codon. And then your amino acid, that is the building block of protein, will correspond to that. So it's got landing gear that is complementary to this. And the landing gear has to be, in this case, CUG. Okay, and that's going to be aspartic acid that it carries. And that's the building block. However, because it's now GGC, it becomes glycine. And because the building block of the protein has changed, it means that they cannot make the surface proteins that bind to the Velcro that comes out of the vessel wall. And literally, it is one A that was changed to a G at the nucleotide position 383. So if you start at the beginning of the gene and you count bases, at 383 there was supposed to be an A, there's now a G, and it sits on chromosome 1. So this resulted in an amino acid change from aspartic acid to glycine at the position 128. Okay, again, sounds weird, but remember, position 128 is the 128th amino acid, not base. These are the bases, this is the amino acids in the CD18 protein. Okay, so again, it's a single nucleotide polymorphism. What the heck does that mean? Single means one base, okay? 1A or 1G or 1C or 1T. Nucleotide is A, T, G, C. Remember, they always bind to the complementary base pairing rule, A to the T, G to the C. Polymorphism. As I've said to you many times, poly times I've said this. Poly means many. Morphism, if you're able to morph into something different, you can change shape, can't you? Okay, so polymorphism means there's many shapes of this single nucleotide. Okay, polymorphous is Greek. It means many forms. So how many forms can a single nucleotide have? It can have 4. An A, a G, a C, or a T. So there's a one base change. That's what a SNP is. So if we look for this one base change and it is present, then it means that our gene that we're looking for is present. Does that make sense? So our marker is just one. It might be the one, like in this case, that causes the problem. Sometimes it's not one that causes a problem. Okay. Then we have SNPs as markers. So as you can see here, AGT. So to be classified as a SNP, the change needs to be found in at least 1% of the total population. Otherwise, it's just a random mutation. Okay? It needs, and on the next slide, I will show you a little bit more detail about that. So if we find them, we can actually see that they are there. So if you look at this picture here, you can see that in this specific SNP, let's say we're looking at this yellow one here, okay? That refers to a T, a thymine. And you can see that 1% of the population has a thymine there, 25% of the population has a guanine, 34% of the population has a cytosine, and 40% has an adenine. So if it was less than 1% for any one of these, then it's just a mutation. It hasn't been established in the population yet. It can only be there if it's established in the population. So they're not always located within genes. They do not always affect the way a protein is used. So linked SNPs, also called indicative, I indicate. They are not within the genes and do not affect the protein function, but they do correspond to a particular allele. So in other words, it can be outside these SNPs, but if they are there, it means that the gene you're looking for is downstream of it. It can sometimes be in the area, the promoter zone, that turns the gene on and off. And if it doesn't turn on, you're never going to have this gene expressed. So that might be the mutation that is happening because of this SNP. Otherwise, it can be in the gene sequence itself. So we have these called causative. Non-coding changes the amount of protein produced. Coding changes the amino acid. So coding SNPs located within the coding region of a gene changes the amino acid sequence. Like we said in BLAD, it was changed and the amino acid changed. Non-coding SNPs located within your gene's regulatory sequence may be turning it on or off. Okay, so you can see this one is in the regulatory and this one is in the coding region. So this blue part is the part that gets turned into the protein. This reddish brown part is not. Okay, so it's going to be a single nucleotide that was changed, and because of that change, we know the gene is present. That is a SNP. Then we get to microsatellites. They are a simple sequence of repeated nucleotides. Okay, repeated nucleotides. So if you look down here, it might be like GCC, GCC, GCC. Can you see that it's a 3 nucleotide repeat? GCC. It can be a 2 nucleotide repeat. CA, CA, CA. And these might be around. So if you look at this, let's say you're looking at this piece of DNA here, and you have CA, 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 and you find something like this, and it's close to the gene of interest, then it might be that if this is present, then your gene of interest is always present. And then you will look at other organisms and see, do they have the gene of interest, and do you find this microsatellite around them, yes or no? And if they are, if this microsatellite is associated with your gene of interest, then you know that you can look for only that. Otherwise, you look for that if it is with the, if it's a gene that you don't want or something. But that is basically how it works. You have your gene either downstream or upstream, and then these things will be close by. So you look for them because it's easier to find them, and then from there it shows you where the QTL is. So microsatellites, also known as simple repeat, are short repetitive DNA sequences that are used as genetic markers. The repeated sequences are usually between 1 and 4 base pairs in size. The specific number of repeats identified to be associated with your allele, if present, then your allele is present. Okay, so if they are there, then your allele that you want is there. So a microsatellite in this case shows up here between 100 and 200 base pairs. So that means this is the allele we want. This one does not have the microsatellite, so it's not the allele we want. This one does have the microsatellite, therefore there is the allele we want. So we can use all these H's and use them to breed with. Okay, can you see this one is strange? But, oh wait, that's the parents. Parent 1, parent 2. Okay, hope that makes sense. So that's where I'm going to stop this video. In the next video we'll talk about, so if we talk about all these SNPs and microsatellites and so on, and they're all base pairs, how do we actually figure out what the sequence is? Okay, thank you for your attention. I shall speak to you again soon. Bye.